Hi, everyone, and welcome to today's lecture on bacterial genetics, which happens to be a combination of two of my favorite babies, which are genetics but also microbiology. So, when we talk about bacteria and study their genetics, the first thing that's important to know about bacteria is actually a little bit about how they're going to grow. That's important in a laboratory setting, but also in things like your body or on surfaces. And bacteria in a lab or in a medical setting, they can be grown in liquid or on agar, which are kind of look like gelatin or jello plates. And there are two categories I want you to know in terms of describing bacteria based on growing in that liquid or on that agar. The first one is prototroph, the second one is auxotroph. If a bacteria is a prototroph, that means that they can grow on minimal media, okay, because they have the ability to make their own nutrients. So you can grow them on a plate or in liquid that really doesn't have much inside of it. Whereas if it is this term, an auxotroph, you have to give them a plate or media that has all the necessary nutrients for a cell because those bacteria have lost their ability to synthesize their own nutrients. So you have to make sure that they have it in their media that you're growing them on. Now, no matter where they're growing, you'll usually be able to describe their growth curve as four main phases. The first phase is called the lag phase. In the lag phase, they are basically just setting themselves up. They're starting to do things like preparing enzymes for DNA replication, for transcription, for translation, but they are currently metabolically inactive, meaning that they are not dividing yet. They then go into the log phase, which you see here is exponential growth. Now they're metabolically active and they are dividing by what's called binary fission. And we're going to go over that in a little while. This is the stage where you can start targeting them with things like antibiotics or drugs, okay, because now they're metabolically active and they'll be able to take in that drug and end up having it kill them. They then get to the stationary phase where the growth flatlines. And if you think about it, if they're growing, let's say in a tube of liquid on your lab bench, well, as they're growing and replicating exponentially, eventually it's going to get kind of crowded. Okay, there's going to be a lot of bacteria and they will have used up a lot of the resources and they're starting to produce waste. So at this point, their cell death rate is going to start to become equal to the cell division rate. So that's why you see it flatlines. Then what happens? Very dramatic, the death phase. And the death phase, you're going to see a steep and steady decline in the amount of bacterial cells present because now too many wastes have accumulated and the resources have run out. So now the cell death rate is much greater than the cell division rate. So you end up having a lot of death and not a lot of new growth. So, boom, the population declines. Now, when we start to think of their genetics, there's a few big differences between bacterial, which is prokaryotic genomes, and eukaryotic genomes, which are our own. Okay, prokaryotes or bacteria, they tend to have haploid and circular genomes, okay? And they also have circular DNA beyond their genome called plasmids as well a lot of times. Whereas eukaryotes, they're diploid in their genomes and they are linear. Okay, so haploid and diploid are what we see in our other lectures where you see N versus 2N. And then circular and versus linear is what I also want you to remember because that'll play an important role when we talk about some limitations to eukaryotic genomes and things like our own aging because it's linear and gets, eventually shortened and shortened. Now, There's something called unidirectional gene transfer in bacteria. You see that over here in this kind of image here in things like conjugation. It's unidirectional, meaning you have a donor giving DNA to the recipient. And you don't have two cells sharing genetic material to create progeny, okay? Instead, it's just one giving DNA. Notice this donor did not get anything in return. You don't have a mixture of the genes in that way, as opposed to if you have two gametes fusing and sharing their genetic material to create progeny. Now, a moment ago, I mentioned binary fission for bacteria. Okay, what happens in terms of binary fission? That's cell division. That's when you have asexual reproduction, meaning you just have one cell undergoing cell division to become two cells. There's no reshuffling of genes or any kind of variety there. Okay, the progeny or the daughter cells will be identical to the original parental cell. Of a lot of great value to us is that with bacteria, what's called gene transfer is also possible in terms of getting new genes or new DNA into bacterial cells. Okay, this is very significant because that allows for genetic variety. It gives us a lot of power also in research world and the medical industry that we'll talk about in later slides and later lectures. Now, there are three forms of bacterial gene transfer that I want you to know. Basically, three ways that bacteria can obtain new DNA. Okay, the first one that I'll mention is conjugation. That will be cell to cell contact, and we call that bacterial sex. In this figure, I should have swapped around the figures because I always introduce conjugation first. Conjugation over here, you see a bacteria transferring DNA to another bacteria through direct cell contact. So the new DNA is coming from another bacterial cell. There's also transduction, which is this middle one here. In transduction, you have a virus vector, a virus inserting DNA into the bacteria to give that bacteria new genes. The first one up here is transformation, where you have free floating molecules of DNA in the environment, and the bacteria can pick that up. Okay, this is different here from this bottom one because notice it's not a direct transfer, it's not cell to cell contact the way conjugation was. Here you have free floating pieces of DNA currently not in any cell that get taken in by the recipient bacteria. Okay, so remember it can either get gene transfer from the environment here, from a virus, or from another bacteria. Okay, remember those three ways. Okay, so now once you've had gene transfer, meaning once you've had new DNA enter the bacteria, there's actually three possible fates for that DNA. The first one here is that that DNA can basically stay as a separate DNA molecule and still do good things for that cell. The second thing is that it can be destroyed in the new cell, so broken down and kind of ultimately excreted. And the last thing is that it can be integrated into the bacteria's chromosome. Okay, so in that case, it's now part of the big main DNA of that cell. It's not a separate little plasmid anymore. Now, for the first one, if the DNA is going to remain a separate DNA molecule within the cell, it has to be what's called a replicon. So, for instance, like these little round plasmids that you see over here, and it needs to have an origin of replication. basically so that that little ring of DNA can get maintained in the population. It can still keep replicating and carry on in that cell. Okay, but it's separate from the big main chromosome. Another thing that can happen, like I mentioned before, is that it can get destroyed. Okay? It can get destroyed in the new cell, especially if it has been a linear piece of DNA that has come in. So, for instance, when you saw the picture of transformation, you noticed some of those pieces of DNA were actually little linear fragments of DNA, and those can get destroyed by things like endonucleases. And so linear pieces or linear fragments of DNA are going to be the types of DNA that's most susceptible to getting destroyed. And again, that we see in humans, we're going to talk about in later lectures, the idea of aging and telomeres because we have linear DNA. The last option is integration into the bacterial cell. It can get integrated if, for instance, it's what's called transposable element or has inverted repeats. Basically, if it has sequences that allow for what's called recombination. So that separate DNA can basically get cut open. The bacterial chromosome can get cut open, and sequences recognize each other and kind of stick together. And we'll talk about that in more detail in later lectures. So now I want to give you a little bit more detail in terms of some of the forms of gene transfer that we just talked about. So the first one I mentioned was conjugation, which I said is basically like bacteria sex. You have two bacteria coming in direct contact with each other, and in a moment we'll go over the exact steps. But the first thing I want to show you is how exactly they discovered that conjugation was actually a thing and that you needed to have bacteria come in direct contact with each other for this kind of gene transfer. So basically, what they did is the experiment that you see here. And what this experiment is showing is that they took two different strains. And what's different about these strains is the media, the nutrients that they require. Okay, so earlier I mentioned how some bacteria can synthesize certain nutrients and then others need to be given those nutrients. When you see a minus sign next to a nutrient or an amino acid, it means that that bacteria is not capable of synthesizing that particular need. And so you have to give it to it in the media, otherwise it will die. So the strain here and the strain here, they're each missing some of the required nutrient abilities, the ability to synthesize some of their required nutrients. So if you grow them on minimal plates, they're not able to survive. Okay, there's no growth on these plates because they cannot synthesize the nutrients that they need. But you'll notice that they have opposite needs. So this one can synthesize three of those nutrients, whereas this one can synthesize the other two, but not those three. When they're combined together, suddenly you're able to see growth on the minimal plates. So it tells you that they have transferred their genes so that now they're able to synthesize all of those necessary nutrients and grow on the plates that they couldn't grow on individually. When you do the same experiment, but you put a special filter between the two cells where only the media can come in contact or move back and forth, but the cells are kept separate, you end up having no growth on the plates. Okay, they need direct growth in order to basically gain new genes, gain new abilities, and in this case, synthesize the nutrients that they would need. So now, how are they actually getting these new genes in conjugation? Well, we already said they have to have cell-to-cell contact, and the way that they're able to have that special contact is through what's called transferability. So they have to have a special plasmid, which we call an F plasmid for fertility. Okay, F for fertility. And that F plasmid will have what's called TRA genes. So basically, they're transferability genes. And what happens is these genes will code for that donor cell, that cell that has the fertility plasmid, we could call it F positive. That donor is now able to form what's called a sex pilus, which is like a lasso to bring the cells together, to pull a recipient close to it, and then the TRA genes also code for that sex pilus to become a conjugation bridge to transfer the DNA to the recipient cell. Okay, and then ultimately you're going to see in the next figures, rolling replication is then able to occur. Okay, so you have the sex pilus and the conjugation bridge being able to be formed thanks to TRA genes. So I want you to circle star highlight the term sex pilus and conjugation bridge in your chalkboard notes on the next slide. So, to help you visualize what we were just talking about, you have a donor cell here, an F positive cell. You notice it has that little transferable plasmid in it, that small circle. That codes for it to produce the sex pilus here, which grabs a hold of that recipient cell. Notice right now the recipient does not have a plasmid, doesn't have that little ring. Okay, so the donor grabs a hold of that recipient. I know it sounds kind of kinky, and it pulls it nice and close, and then the TRA gene codes for the sex pilus to turn into a conjugation bridge. Okay, when that happens, the double stranded plasmid gets nicked. What nicked means is one strand of it gets cut, and so that single strand starts to unroll here. Okay, so it's separating from being double stranded. That single strand that you see here crosses through the conjugation bridge and enters the recipient. Now you have a ring of single stranded DNA in the donor, a ring of single stranded DNA in the recipient, and they can use that to have as templates for replication. So they turn those single stranded DNA into original double stranded DNA again. So now both the donor and the recipient now have the plasmid. So when we talk about F plus and F minus cells, the donor originally is F plus, it has a plasmid, the F plasmid. The recipient was F minus. It did not have the F plasmid. At the end, what do you notice? The donor still has the plasmid. That's why it was important that it was just a single strand transfer. The donor still has the plasmid. It's still F plus. But now what's the recipient? It is also F plus. Please star that in your notes, okay? Make sure to write that down on your notes here that originally the donor was F plus, F as in fertility, okay? Donor was F plus, recipient was F minus. At the end, the donor is still F plus. Now the recipient is also F plus. They both have the double-stranded plasmid at the end, okay? Because they used the single strand as a template to synthesize the proper double-stranded plasmid. Okay, and then I just threw in this fun fact to scar your minds. Yeah, conjugation tends to be a cluster, so these are actually three different cells. And yeah, so it's basically bacterial orgies. So think of that when you think of your microbiome and the bacteria that are in your gut and on your skin. They're totally having nice little sex orgies on your skin and in your body. Yeah. Enjoy that visual. So now that I've traumatized you and ruined your mindset every time you're looking at your arms or take probiotics, the next thing I want to mention is we said that plasmids and little fragments of DNA are getting transferred in these gene transfer, examples that I gave you. But sometimes chromosomal genes can actually end up transferred as well. So usually, sorry, let me get the laser pointer. Usually you're transferring plasmid genes, but sometimes chromosomal genes can get transferred as well if the plasmid integrates into the chromosome. So if this DNA integrates into this big DNA here. And the way that it can do that is basically if you have what's called IS sequences on both the chromosome and the plasmid, those are insertion sequences. Again, this all sounds very dirty for bacteria. So they have to have an insertion sequence in the chromosome and an insertion sequence in the plasmid. Okay, and that will allow for recombination. That's basically a recognition site for a recombination protein. So that recombination protein will come along, it will cut open the plasmid, it will cut open the chromosome, and it will link them together. And so now you have both the plasmid genes and the chromosomal genes. Now, since the plasmid genes code for conjugation, okay, because it's an F plasmid, so it induces conjugation, what happens now is when the F plasmid genes code for conjugation for that pilus and the conjugation bridge, notice that it's now going to transfer all of this DNA. So that includes chromosomal DNA. Okay, so you then end up allowing for a whole lot of genes to get transferred. So now that was conjugation. So to bacteria, direct contact transfer of DNA. We also mentioned that transformation is possible, and this one you tend to cover more in your laboratory classes in a lot of detail. So we'll just briefly go over the general idea. Basically, again, transformation is when you have a bacterial cell taking up naked DNA. So that's DNA fragments that are in the environment around the bacteria. They're not currently in any other cell. Okay? In order to take in that free floating DNA, the bacterial cell has to be what's called competent, unlike a lot of my students. But anyway, competent in terms of the cell means able to take in that DNA. Again, everything I say sounds so inappropriate, but yeah, able to take in the DNA, and we're going to see some examples of that. Basically, think of it as the outside of the bacterial cell becoming more porous, you can think of it as. Okay? There are three ways that that cell can become competent and ready to take in that DNA. One way is called naturally. The way that you tend to use in laboratories is chemical competence and also electroporation competence. So, some bacteria, if it's naturally competent, that means they don't need treatment, they don't need anything, they could just naturally take in that DNA. If it's chemical competence, what we do is we put it in solution, like a cold calcium chloride solution, and you basically have it mixed with metal ions a lot of times that basically allow the cells to undergo a membrane change to create an ionic bridge. These are details you won't have to know yet. I'm just kind of giving you a general idea. And so they're then able to pull in negatively charged DNA because you've changed their outer membrane to a more positive form. And originally the outer membrane of the bacteria would be highly negative because of all the lipopolysaccharides on it. So that's chemical competence. And then electroporation is when you use high voltage to basically zap open the cell wall. Okay, all of those techniques you're going to go over more in laboratory courses. Now, what's interesting is that transformation, that ability of bacteria actually helped us to know that DNA is actually the genetic material of cells and that it's not proteins, for instance. Because, a lot of times before we really knew what DNA was, they weren't sure what actually was the genetic material that gets transferred to offspring. And so in this figure here, this experiment was done by a scientist named Avery back in 1944. This is before the 1952 Hershey and Chase experiment that we also cover in this class, which is the one with radioactive labeling. But in this one, this was an early way to kind of help suggest that DNA is the genetic material and not proteins. So Hershey and Chase later confirmed it more, but this one suggested it. And so basically what you see here, there are two types of bacteria. There is the smooth variant of the bacteria that has an outer capsule and is highly virulent. Okay, virulent in that it ends up killing a mouse if it's inside of that mouse. Okay, it's highly severely dangerous, you can think of it as. There's also the rough version, the rough variant. It lacks that capsule around it. And this rough version ends up not being highly virulent. Okay, it does not kill a mouse. So, what they ended up doing is they purified DNA from this smooth, dangerous bacteria. So, they purified just the DNA out of it. They then took that pure DNA and they injected it into the environment around the bacteria. Okay, so they are now taking pure DNA and they are putting it in the environment around the uh, non-virulent bacteria. Okay, and what happens was this non-virulent, you can think of it as a safe, not dangerous bacteria, suddenly took in the dangerous DNA. It then was able to produce the dangerous characteristics, such as that capsule. So now it took on the smooth colony's characteristics. And now, if you transferred that one into the mouse, now the mouse would die. Okay, so it showed that the transformed bacteria basically took on the characteristics of the bacteria where the original DNA came from. Okay, so this rough DNA, this rough bacteria did not have the DNA to make a capsule or to kill a mouse. But when it was put in an environment with just the DNA from the bad guy bacteria, now it was able to do all of the same things that the bad guy bacteria could do. So it showed that you only needed the DNA and not the protein, not any other part of that cell to carry on genetic information, okay, to give the traits to inherit new qualities. So now, since in genetics lab class that goes along with this lecture, you tend to cover, transformation, having chemical competence or electroporation in a lot of details. I don't really get to include that in the lecture portion. What we do cover instead is natural competence that you don't usually use in the laboratory. For this one, what you notice is you have this long piece of double stranded DNA. It gets recognized by a receptor on the outside of the naturally competent cell. And then what you notice is next to that receptor, you have endonucleases and exonucleases. Okay, so the endonucleases will chop up that DNA Okay, so now you're getting smaller and smaller fragments. And then the exonuclease will then degrade one strand of that double-stranded DNA. Now that it's a nice small fragment and it's only single-stranded, now that's able to get taken into the cell and into the cytoplasm of the bacteria. Now notice when it's natural competence, you're only taking in a short fragment and it's single-stranded DNA. This is very different from when we do laboratory experiments, like I mentioned, the ones that you'll get to do in the lab class that we have. In those, you're doing chemical competence, you're doing electroporation, those artificial transformation methods, you're able to get large amounts of double-stranded DNA into the cells. Okay, because basically you're forcing damage onto the cell wall, or you're forcing, the cell to accept a larger piece of DNA. Natural competence, though, has the limitation to please remember it's just short single strand of DNA taken in. Okay, so we talked about conjugation, where bacteria can get new genes, new DNA from other bacterial cells. We talked about transformation, where they can pick up new genes or DNA from the environment, so naked, free floating DNA. The last one I want to mention today is transduction. And so in transduction, there's going to be a virus. And when I ask here what type of virus, the answer is a bacteriophage. Okay, bacteriophage. I'll have that spelled out for you on the next slide for the chalkboard notes. But I want you to circle, star, highlight that term when you see it on the next slide, bacteriophage. That is a phage, so a virus, that infects bacteria. And it looks like a little space creation, okay? And it injects its DNA into that bacteria. Now, once that DNA is inside that bacteria, you could end up getting some recombination occurring where you get a mixture, you get some bacterial DNA, some bacterial genes that get packaged into new viruses. When those viruses then go out of the bacteria and infect other bacteria, they can now give the next bacteria some of the original, the previous bacteria's genes. Okay, so that can end up being very valuable in terms of genetic variety because you have these viruses taking DNA from one bacteria and transferring it into others. Now, when we say that they can transfer bacterial DNA, there are actually two forms of transduction, and they can be generalized or they can be specialized. So basically, in generalized transduction, when you have bacterial DNA ending up packaged into the viruses, in generalized transduction, the bacterial genes that get packaged are basically random. Okay, so if you have a bacterial chromosome, any of the genes have equal probability of getting transferred into the virus if it's generalized transduction. But a lot of times, certain bacterial regions will end up transferred with preference. So we call that specialized transduction. Okay, so when you see here, there are certain areas of this chromosome that is more likely to get packaged, and then ones further away from that region are less likely to get packaged. So um, it if you look at this lambda DNA, it's going to show you that will be the most frequent packaging. And basically, the lambda bacteriophage integrates the phage DNA into the bacterial chromosome. And so then when that's eventually getting excised or cut out of the chromosome, only bacterial genes near that DNA will get transferred. So that's why this area would be very unlikely to get transferred in specialized transduction when you're dealing with something like a lambda bacteriophage. Okay, so only the ones closest to the virus DNA will end up transferred. So that's the difference between generalized versus specialized. So in this case, the gal and the ATT, they're closer to the lambda, so they'd be more likely to get transferred than any of the pink region that we didn't even label. Okay, so that's enough of gene transfer. There's just a couple of other things I want to mention before we finish this lecture. When we talk about bacteria, I want to make sure you know the difference between gram-negative and gram-positive bacterial cells. And this is something you cover in a ton of detail if you take microbiology, and it's very important if you're going into the medical field. So, when you look at gram negative and gram positive cells, I made these little diagrams on the right. When you look at the gram cells, they're basically differentiated based on differences in their cell wall. So they're called gram negative and gram positive based on how their cell wall is composed. This one up here, I want you to circle and label as the gram negative cell wall. When you look at this, you notice that there are a lot more layers to it. There are a lot of lipids sticking out of it, and there's what's called a highly selective porin, which is like a little channel that allows things to enter or exit that cell. Okay? All cells, for our purposes, there are examples that don't meet this, but for our purposes, all the cells have the plasma membrane. And then on top of that is what differs. And so you notice in gram negatives, there are a lot more layers on top of that plasma membrane. The first one being a very thin layer of peptidoglycan, whereas the gram positive over here has a very thick layer of peptidoglycan. That's basically a heavily carbohydrate matrix that has some peptide groups mixed in as well. And when we look at this, you notice that beyond that, on the gram negative, you then have the extra layers. Now, when you look at the gram positive, you don't have all of those extra layers. You don't have the selective porins. What's sticking out here is teichoic acid, and if you take microbiology, that's important in terms of infecting people and being toxic to people and causing symptoms in people. But for our purposes, one of the questions I ask here is which do you think is tougher to treat with antibiotics or drugs? A gram negative or a gram positive cell? So think about it. If you want to treat someone who's suffering from an infection from bacteria, you want to get that antibiotic or drug into that bacteria. When you look at these two cells, what do you notice about the gram negative? Psh, that's a lot of different layers, and it has highly selective porins on that outer membrane, that extra shell. So it's going to be a lot tougher to get antibiotics or drugs into the gram negative. So they will be tougher to treat. On the opposite thinking scale, when I ask which is better for genetic engineering, well, now think about it. For genetic engineering, you want to be able to easily get things into and out of that cell, right? Which one of these are you going to easily get, for instance, new DNA into? Well, that would be the gram positive, right? It does not have all those extra layers. It is not as highly selective with those porins, okay? So for genetic engineering, you're going to want to pick, if possible, a gram positive. It will make your life a little bit easier, okay? Whereas you notice in terms of the medical field, when we talk about problems with treating gram negative versus gram positive, gram negative, I want you to remember for this first answer here. The very last thing I want to mention in this lecture is that way back in the day, we originally had two categories of bacteria. They were called eubacteria and archaeobacteria. So eubacteria is all the bacteria that you guys know of now. Okay? Things like E. coli and Staphylococcus. Okay? Those are all eubacteria. Archaeobacteria, what those are basically bacteria that were able to survive in extreme conditions. Okay, they would lack the kind of peptidoglycan walls that we just showed with the gram positive and negative, and they are very hardy types of bacteria. Okay, now, or very hardy types of organisms. Now, when we think of these, since I'm saying that they can survive in extreme conditions and they're very hardy and they would be able to live in places like thermal vents or a volcano, when you think about that, that tells you something significant about the archaea. That tells us that they would be very valuable to us because if they're able to survive in extreme heat, for instance, extreme pressure, extreme salt content, these kind of very strenuous extreme conditions, that means that they have enzymes that produce proteins for them that allow them to survive those conditions. And so we can extract those, we can use those. So, for instance, when you perform your experiments in lab, you'll notice when you do, for instance, PCR, you'll hear Taq polymerase. Well, that when you do PCR, that has to survive or not survive because it's not living. That has to not be denatured at very high temperatures because we put that PCR up to, for instance, 95 degrees Celsius. So you need a hardy protein or you need hardy enzymes. And so we use ones that we extracted from archaea that were able to survive in those kinds of conditions. So that's very, very valuable to us. But what's interesting is, even though they called them both bacteria, they called it eubacteria and archaeobacteria, the two of those are actually no more related to each other as either is related to eukaryotes, which are us. Okay, so they are not closely related, not the same thing, even though they both, you a lot of times would hear them called bacteria. So, just to see how you're paying attention, here's the review question for this lecture. Please send me the answer in the Remind app, or if you are not part of one of my classes but you still want to participate, you can use my email address that's listed on my channel and send me the answer of which of the following would be most difficult to treat and why: a gram positive infection or a gram negative infection. And that is it for today. If you enjoy things with bacteria, I highly recommend taking courses in microbiology or virology. Very exciting, especially after you have lived through a major pandemic. All right, that is it for today. Thank you and have a great day.